Now let's talk a little more about the cell. We will talk only the most important MCQ points. Coming to the cell membrane. The cell membrane is classically given by the fluid mosaic model. Here the fluid part is nothing but our lipid and the mosaic part is nothing but our protein. This one common mistake everybody does, which is the maximum composition of the cell, whether it is a lipid or a protein. All of us know that cell is a lipid bilayer, but that doesn't mean that lipid is more. Even though the cell is a lipid bilayer, still the protein composition is more. This has been a repeat MCQ and repeat made mistakes by the students. Protein is a major component. Just because it is a lipid bilayer doesn't mean that lipid is maximum. But when you consider the weight, it is almost 1:1 ratio between the protein and lipid. There are some exceptions which we have to know. Here one important exception is only one area the lipid content is very high. This lipid content is present maximally in the myelin sheath. The myelin sheath is the one which is myelinating the neurons. There the lipid content is more. Just remember this. And there is maximum content of protein which is present in one region, that is the inner mitochondrial membrane. Protein is usually higher, but maximal concentration is present in the inner mitochondrial membrane. So these are two MCQs which can be asked. So coming to the cell cytoskeleton. What is cytoskeleton? Skeleton means bones of the body. There are few things which are acting like a skeleton to the cell. They are called a cytoskeleton. They help to maintain the shell shape and size. The first one is microfilament. Microfilament, all of us should have studied actin and myosin. They are the microfilaments. The primary unit is made up of actin and it is the maximally available protein. This is the maximally available protein. And its function is contractile protein. Actin and myosin will study in the muscle physiology. It is a contractile protein. Then comes the intermediate filament. The intermediate filament is made up of several proteins, but it doesn't have any single primary unit. But they are used for cell markers. They are primarily used for cell markers, especially the keratin and vimentin. Both of them are used for the cell markers. Then finally comes the microtubules. The microtubules are like the roadway inside the membrane. If there is a cell, there is a microtubule. All the molecular motors will walk and travel in this microtubules, over this microtubules only. It is like a road inside the cell. Its primary unit is tubulin, and the molecular motors which are primarily involved is kinesin and dynein. Kinesin and dynein. So both of this we will study in the nerve muscle physiology extensively. They are very, very important. This kinesin and dynein. What is the function? They are moving the substances between the different parts of the transport. We will study about them in the axonal transport in detail. Then comes the cell to cell junctions. Two cells are connected with the help of various junctions. That is the cell to cell junction. The primary junction, where you can see here, there are something called as tight junction. What is the function of these tight junctions? Tight junctions, they tightly bound the two cells. This will be helpful in some places where the barriers are there. What are some barriers? Just name them. There is some blood-brain barrier. There is blood-testis barrier. All these areas need to protect the entry of most of the molecules. So that's why we have tight junctions in these barriers. So that is the reason these are called as tight junctions. And it is also called as zona occludens. Occludens means it is creating some occlusion. That's why it is called zona occludens. It is also made up of claudin. The proteins involved are claudin and occludin. The protein name is important. Claudin and occludin. Occludin we can remember with the name of occludens. Then comes the desmosomes. This is also a cell to cell connection. In desmosomes, what will happen is it is also putting some strong adhesions between the cells. That's why it is called zona adherens. What is the difference between a desmosome and zona adherens? Desmosomes will have intermediate filaments which is attaching them. So it is basically strengthening the cell. In our cardiovascular muscle, we will read about these desmosomes which are called as intercalated disc. They keep the cardiac cells close to each other and prevents the overstretching. So this intermediate filament and desmosomes are present in there. Now comes the final group of cells which is connecting the cell to cell junction, that is the gap junction. These gap junctions, what they do is, if two cells are there, there are some gap junctions. These gap junctions, they will connect the cytoplasm of one cell with that of the other cell. It's like both the cells are connected to each other. This form of connection is especially important in cardiovascular system, wherein we will be studying about the syncytial activity. Syncytial means all the cardiac muscles, the atrial will contract together and the ventricular will contract together. This atrial and ventricular syncytium is possible only because of the gap junctions. And the protein involved in formation of them is connexins. This connexins is the protein involved in formation. Then there are some junctions between the cell and the basement membrane. There are two important ones. One is focal adhesion, another one is hemidesmosomes. Hemidesmosomes looks like a desmosome, that's why it's called hemidesmosome. It is made up of a protein called integrin, whereas focal adhesions are made up of actin filaments. So these are the cell to cell communications connections you should know. These are very, very important because any of them can be shown in pictures also, and some of it is associated with the syncytial activity like gap junctions, and the connexins is a protein for them. So this is very important. Now coming to the intercellular communication. This intercellular communication, recently it was asked in an MCQ also. Gap junction we have already discussed. It is just like attaching two cells and there is a protein which is transporting substances between these two. Second thing is synaptic cleft. This we will study in the neuromuscular junction, where there will be a connection between the neuromuscular junction, that is the synaptic cleft. Neurotransmitter will be formed here and it will be sent to the other side, and there will be receptors on the other side. Then autocrine. What is autocrine communication? This is not intercellular actually. This autocrine means it will have a product which will act upon its own receptors, that is autocrine. Self-activating receptors will be there. Then comes the paracrine. Para means neighboring cells. A substance is produced here. If it is acting on its neighboring cells, then it is called paracrine. Recently this was shown as a diagram and asked as a question, what is the phenomena observed here? It is a paracrine action of the neurotransmitters and action of the hormones. Then endocrine. Endocrine, all of us know, whenever any substances is produced in one side and it is transported in the blood, and if it is reaching its end organ, then it will be called as an endocrine kind of communication. So intracellular communications, since they have asked paracrine, they might ask the endocrine or synaptic cleft or autocrine the next time. They'll give you a picture which is simulating on its own, then it is autocrine, and you know the other functions also. This neuromuscular transmission we will again study in the neuromuscular disorders also.